Welcome everyone. We'll just wait a few minutes for others to join in. We still have people coming in. Good afternoon, Dr. Swilian. Thank you for joining us. I'm doing fine. So we have with us more than 100 participants, and we were expecting from 40 over countries. We had a very high registration, and we hope they will join in, but those who are not with us can certainly view the recording later. So welcome. Good morning, good afternoon, and good evening to everyone. And for those who are staying up at odd hours, we hope that the webinar will certainly keep your mind awake, because we are going to talk about something that's really important and which affects public health, especially in tropical countries. So this is our third webinar on Gene Drive, organized by ISA. ISA is International Service for the Acquisition of Agri-Biotech Applications. And it's also organized by Outreach Network for Gene Drive Research, and together with ISA partners, the Biotechnology Information Centers around the world, especially in Asia, and by CELCA. This webinar is on Can Gene Drive Eliminate Vector-Borne Diseases? I am Dr. Mahalakshmi Arjunan, Maha for short. I'm the global coordinator of ISA, and I'm trained in biotechnology and microbiology and have a PhD in science communication. Before we start, here are a few housekeeping rules. If you have any questions, today we are using the Q&A function. So please type your questions there and we'll pick that up for there. Keep yourself muted, and this will also be done by my team at ISA, and your cameras off to reduce the interference for us to have a strong internet network. And also, a lot of people usually ask if you're going to get a certificate. Yes, you will, but you have to respond to the post-webinar survey, and you will have to be with us until the end, because the link to the survey will be posted at the end of the webinar. We will try as much as possible to end the seminar in the time stipulated. So please stay with us till the end. Speaker's presentation in PDF will also be posted on ISA's website. Please visit our website. It is isAAA.org. You will get much more information there. My colleagues will also type our website address on the chat box. The recording of previous webinars will also be there. So there's very rich information there. A little brief information about ISA. We are an international not-for-profit organization, and we champion knowledge transfer and capacity building on biosciences, policies, and regulations. Gene technologies is certainly one of our core areas, because this creates a huge impact potential, but it also causes a lot of debate and controversies. And today we are working on gene drive, and this is definitely going to be a focus area for ISA, because we see this as a new technology with potential in many areas such as biodiversity, public health, and in controlling agricultural pests, which is also our interest. It is important for stakeholders to come together and discuss technology such as this, so that it can be advanced responsibly and the myths and concerns can be addressed. Allow me to now introduce the moderator for today's webinar, Professor Sheetal Silal. Professor Silal is the director of the Modeling and Simulation Hub Africa, MASHA, and associate professor in the Department of Statistical Sciences at the University of Cape Town. She is an honorary visiting research fellow in tropical disease modeling at the Nuffield Department of Medicine at Oxford University. She received a PhD in mathematical modeling of infectious disease in 2014 from UCT, University of Cape Town. She works on mathematical models on malaria, pertussis, COVID-19, and other infectious diseases in South Africa, Sub-Saharan Africa, and globally, to predict the dynamics and to control of diseases, and to evaluate the potential impact of control programs in reducing morbidity and mortality and supporting policy development. So, of course, she's the best person to moderate today's session. And with that, I passing the screen over to Professor Sheetal Silal.
Thank you, Dr. Maha, and welcome to all participants to this webinar on can gene drive eliminate all vector-borne diseases? So let's get right into it and introduce our expert panelists for this event. First up, we have Prof. Eng Ong Ooi, who is a professor in the program in emerging infectious diseases at Duke NUS Medical School, Singapore. And he co-directs the Viral Research and Experimental Medicine Center at SingHealth, Duke NUS Academic Medical Center, that's Viremics, which aims to develop molecular endpoints for use in viral disease therapeutic and vaccine trials. Second on our lineup today, we have Dr. Chun Hong Chen, who is an associate investigator at the Institute of Infectious Disease and Vaccinology of the National Health Research Institutes of Taiwan. He has also been assigned as the Deputy Executive Secretary in charge of mosquito control and novel intervention development in Southern Taiwan, as well as the Chief Executive Officer of the National Tropical Disease Control and Research Center. And our third speaker on our panel in this event is Dr. Maria Vittoria Mancini, a vector biologist specialized in developing innovative control strategies for mosquito-borne diseases. She is a senior researcher at the Polo GGB in Terni, Italy, and she is a part of Target Malaria Project. So I'm sure you'll agree with me, we have quite the expert panel lined up for our webinar today. And let us get right into it with our first presentation with Prof. Eng Ong Ooi. Thank you. Over to you, Prof.
Thank you very much. Thanks for the very kind introduction. And thank you also to ISAAA for this opportunity to be here with and join you on this webinar. So my job is to basically give you an overview of malaria and dengue, and hopefully that frames the context for the next two speakers. And I guess these two diseases are not new to many of us who have heard these terms before. So I'll just take a few minutes just to run through some basics, and then show where perhaps gene drive and mosquito control and all that could make an impact. We know, of course, that there are these malaria is caused by a parasite that is spread by mosquitoes, principally the Anopheles mosquitoes, and depending on which part of the world you are, the species can be different. The most famous of all, those in Africa and in Asia have a different group of mosquitoes, but also belonging to the Anopheles genus that spread these parasites. Now, classically on the left-hand side, we know that there are 4 species of malaria parasites that infect humans, but now we acknowledge that there's a 5th one called Plasmodium knowlesi that originally is spread between the mosquitoes and monkeys, but now it's successfully crossed over to man, and that can cause human malaria as well. This is the life cycle of malaria. So the parasites injected by the mosquito when it feeds on humans for a blood meal. The parasites then get to the liver. I'm not going to go through the terms, but essentially it'll stay in the liver for a while, then it'll emerge from the liver, and then infect the red blood cells. And depending on the cycle, and that goes up somewhere between 48 and 72 hours, they complete one round of life cycle. And because of that 48 to 72 hour life cycle, that's why we get these fever spikes every 48 or 72 hours. And that gives rise to the classical fever pattern of malaria. The parasites then differentiate into the male and female species before they then get taken up by the mosquito for subsequent maturation and then onward transmission. So this is on the left-hand side, it's the map of where the burden of malaria is. And of course, as you can see from the color, it's mostly focused in sub-Saharan Africa. There's Asia as well, that although not as intense a color as sub-Saharan Africa, as well as Central and South America. It's estimated that somewhere between 300 to 500 million cases a year, 1 to 3 million deaths. Most of these, of course, are in children in Africa. And then, of course, because of all these burden of acute disease as well as mortality, that gives rise to quite a big cost to the economies of the countries that suffer from malaria. So, of course, the good thing about malaria is it's a drug. Unlike dengue, there's no drug just yet, and I'll show you what all that means in a minute. But these are the drugs that are in use for for treating malaria. But what you see is that there's the problem with treatment, that there's drug resistance, right? So on the top of the graph, you see the different names of the drug and when these drugs were introduced. And so these are the years. So for instance, chloroquine was introduced shortly after 1945, but resistance to chloroquine was first detected in the late 1950s in Thailand, Cambodia border, and so on and so forth. So as you move along even with the newer drugs, the combination with artemisinin or artemisinin derivative, we're still also encountering resistance developing, especially in Southeast Asia. So this presents problem, therefore, on how we can manage malaria, the global burden of malaria, if the parasites increasingly become resistant to treatment. Just a few months ago, in fact, a few weeks ago, there's an announcement that we might be at least a step closer to a malaria vaccine. And this is the work of the Jenner Institute at University of Oxford, where they've tested their malaria vaccine construct, and it seems to be generating some very, very promising results. Of course, larger trials are needed, and so just watch this space. We're not quite ready for prime time, but at least I think the understanding of the immunology of, or at least the immune response to the malaria parasites now beginning to be sufficiently mature for us to be able to take advantage of what we know to design better vaccines. As you know, many of those in the past have failed and many have dropped off clinical development. So perhaps these ones would be promising enough for us to take to the next stage, and hopefully we'll have a malaria vaccine soon, or as soon as not in the not too distant future. So switching gears a little bit, this is dengue virus. It's spread by different species of mosquitoes from those that spread malaria. In this case, principally Aedes aegypti and Aedes albopictus in our part of the world and in most parts of the world. But in the Pacific Islands, there's a third species called the Aedes polynesiensis that also can transmit dengue virus. On the right-hand side, what I'm showing you here is basically a thing called antigenic cartography, basically how different are these viruses. And so if you look at it, there are actually 4 different dengue viruses called unimaginatively dengue 1, 2, dengue 3, and dengue 4. Now, for those who are interested in dengue, you would think that because they share the same name, dengue virus, and then called 4 different types, that actually there's one species of virus that perhaps 4 different variants, right? If you borrow the terminology of COVID-19, then these are variants. In actual fact, they're not. They are actually genetically more like 4 different viruses. So we're stuck with this system of naming these viruses, and we call them dengue virus and then give them a number, but effectively these are 4 different viruses, so they don't behave in the same way. Now, if you recall in the malaria map, most of the darker colors were all in Africa. Dengue is the opposite. Most of the disease burdens in Southeast Asia and South Asia, as well as in Latin America, right? And so annually, it's thought that there are about 100 million infections, 100 million cases a year of acute dengue, about 400 million infections annually to go around the world. Again, that's probably an underestimate of the true burden of dengue, because a lot of it is probably misdiagnosed as something else. And the reason why it's misdiagnosed is because dengue, if you look at the left-hand side, so right at the top, it just tells you the severity, right? From dengue with warning signs to severe dengue. So if you, and that goes from the left to right. But if you look at dengue, so classical dengue without any complications, you get symptoms like nausea, vomiting, rash, aches and pains, and low blood count and all that, but of course with fever. If I take these symptoms and I say, well, this is actually chikungunya, I would be correct. I could say this is Zika virus infection, and I would be correct. I could even say this is actually COVID-19, and I'll probably be correct too. So it's very difficult to differentiate dengue from many other viral infections, acute viral infections, without lab tests. But of course, what we're not so much concerned about just classical dengue, because people should recover, although about 30% of adults with dengue go on to get very debilitating long-term illness like tiredness and inability to concentrate. But we're of course more afraid of this part of the spectrum, where potentially, if not properly managed, patients can die from these complications. And these are in the form of plasma leakage. So you get low blood pressure, internal hemorrhage, where you bleed into the gut and into other organs, as well as organ impairment, where your liver, your kidneys, and all that start to fail, right? So the idea of treatment is then therefore to try and prevent these severe complications. Now, right now, this treatment is all entirely symptomatic. In other words, if you have low blood pressure, we try and support your blood pressure by giving you fluids. If you are bleeding, we give you blood transfusion. Ideally, there should be a drug that would knock out the virus. And many people have tried. We've tried the various licensed or repurposing licensed drugs or new drugs. Essentially, the end results all over here, none of it have worked. There's nothing that has worked so far. There's one final drug called, this is an antibody that should have started clinical trial, but unfortunately, because of COVID, this trial is now on hold. Dengue vaccine, there is one single licensed vaccine right now by Sanofi Pasteur. Unfortunately, it can only be given to those with a prior dengue infection. If you've never had dengue before, then this vaccine doesn't do you much good. In fact, it may put you at risk of severe dengue. A second vaccine is probably going to become licensed fairly shortly, perhaps later this year or early next year, and that's developed by Takeda. Again, it's promising, but it's not going to work against all 4 dengue viruses. So maybe just a couple of them. There's a third one that's undergoing clinical trial. Hopefully that'll be completed early next year, but it will take another 2-3 years of long-term follow-up before any regulatory authority would license this vaccine. So essentially that's the landscape. But I just want to finish on this note and suggest to you that, despite having drugs for malaria, having vaccines for dengue, that we still need an integrated control measure, because not one single tool can prevent these dengue or these diseases like dengue and malaria. So this is Singapore's experience with dengue control. The red line is the mosquito population density. The blue bars are the cases. So dengue was a prominent disease. You won't realize it, but it actually was because it caused a lot of deaths in the 1960s. And so the Ministry of Health decided that they will control the mosquitoes, right? So which continues up to today. So as you can see in 1966, about 50% of households, so premise index is the number of premises with Aedes mosquitoes breeding in them. And the answer is about 50% of them were positive for Aedes mosquito breeding. But with control measures, this now dropped to, over this period, less than 3%, and right now it's less than 1%. With this initial drop, there became a period where dengue was very well controlled for about 15 years. But since the 90s, we have had these 5-7 yearly spikes in dengue cases, culminating, of course, in last year with well over 30,000 cases of dengue in Singapore, despite the very low mosquito population. And that's the reason for this is because of immunity, right? Because as you control the disease, then fewer and fewer people became infected, and then more and more people became non-immune and therefore susceptible to reinfection and then requiring this disease. So and this is on the back of a highly successful mosquito control program that even with that, we still need a vaccine. So I think the end result is therefore that we need multiple approaches to try and tackle malaria and dengue. In a way, it's like a football game since Euro 2021 is going on. No team goes into a match with a strategy of taking one single shot at goal and hope that goal will be scored and that they will win with a 1-0 score. Nobody does that. So I think the more shots we can take at goal, the better we will have a chance. The better our chance would be of controlling malaria and dengue and reduce the global burden of disease. So I'll stop there and I'll pass the time over to the other speakers. Thank you very much.
Thank you, Prof. Ooi, for clearly laying out the scale, the impact, and also the treatment pipelines for mosquito-borne diseases like malaria and dengue. And you highlight the most important point of how a multi-level, multi-pronged approach is required. And so there is a clear need for innovation in how we combat these vector-borne diseases. So I'd like to then move on to our second expert panelist today, Dr. Chin Hong Chen, to introduce the idea and the notion of what gene drive is, how the technology works, and how this may contribute towards better disease control. Over to you, Dr. Chen.
So can you see the slide?
Yes,
okay.
We can.
Okay, so I would like to thank Isaac for invitations to share some idea about why we need a gene drive mosquito for today. So in this session, I will explain the reason why we need it. And the other, we also will spend time to tell you how to design the anti-malaria or anti-dengue and Zika mosquito in the laboratory. So we all know the mosquito is a very important, also it's a major threat for the public health. So once we want to stop the plague or infectious disease, the better choice is use kind of vaccination. So we have some successful cases, just like smallpox or smallpox or polio, and you can see once we have the comprehensive vaccination plan around the world, and that disease actually can stop. And also from the pandemic for the COVID-19 recently, we also observed some country right now has a more high percentage of vaccination. and also the infection rate will dramatically decrease. But unfortunately, for the vector-borne disease, such kind of the vaccine is not still available, especially not all of them not on the market yet. And we like to thank Dr. Eun Wee's talks and describe a little bit about the progress for the malaria and the dengue and Zika. We do have some malaria, but they also engage some drug resistant. But unfortunately, the dengue and Zika, that kind of the vector-borne disease are actually virus disease, and they are very difficult to find effective drugs so far. So for such kind of the mosquito-borne disease, to control mosquito density will be the key for the action. So we know the malaria actually was transmitted by majorly the genus called Anopheles mosquito. And they actually are more than 100 species could transmit such kind of disease. And there are 2 or 3 major vectors, and the habitat are all located in the sub-Sahara regions. And that actually could cause the million deaths per year. And also that the deaths also most come from the children under the 5 years old. So the other is dengue. Dengue are transmitted by 2 major vectors called Aedes aegypti and Aedes albopictus. So the morphological and they are a little bit different. And for major transmit vector for dengue is Aedes aegypti, but for chikungunya, Aedes albopictus also is very could transmit as well. So you can see the habitat location around the world. In the region in yellow, you can see there are more to only for habitat for Aegypti, and in the blues, it's albopictus. In red, both species are actually living together. So you can see almost around the world, the temperate and also tropical region are their habitat region. So making control for sexual disease a little bit difficult. So what kind of tool we have so far? I would say the chemical fogging actually is the most popular one. So since after the World War II, we have developed a chemical called DDT, and it's very powerful. And some countries actually have got benefit from this invention and to eradicate the malaria disease. But you can see, it's a little bit ironic. You can see another technology like a foam technology. After World War II, since now you can see how dramatic change for this technology is. But for the mosquito control, we seem to be still sticking on the chemical fogging since from the decades ago. So it's really urgent need other tool, and we know the tropical mosquito treatment actually could last only 30 days, but as we know, most of them are less than 2 weeks. So using such kind of strategy, we could find the dengue, which is the vector-borne disease, it didn't been controlled. So from the WHO report, we could see the Asia or America, that such kind of the viral mosquito-borne disease are increasing. So the other way is for mosquito control is the mosquito source reduction. Such kind of the activity is require a lot of the healthy bureaucracy and the good public power to enforce it. And people have to clean up all the vessel around their household, and sometimes need to change the environment condition, just like drain the water body around the area. And they also cost a lot of labor and money as well. So the huge manpower mean huge economic costs. So in some metropolitan city, we usually could find to correct such kind of manpower, sometimes need to have like more than 10,000, maybe more for such kind of activity. And those kind of activity need to be sustained for a long time. And most of the time, it's very difficult to contain and achieve that. So that's why I call this is Sisyphus style control, because we know the story of the Sisyphus. He always forced him to roll in a huge rock up to the hill, just like a chemical forking or the activity we done. And every time when he need to the top of the hill, then the rock always rolling down. So he had to repeating this action forever. So both kind of this work are actually will spend a lot of labor and sometimes it's useless. So we really need some new tool for preventing vector-borne disease. So for mosquito control, there will be two categories of method. The one is we call it the population suppression. Just I mentioned earlier, the chemical forking is one of choice, and also source reduction is also one of these. And if you could repeat in doing such activity, you could see the population actually could be suppressed. But you have to repeatedly, maybe in the whole lifetime, to do this activity. And the other is a biological control method. That could be a little bit easier, and that one is called SIT. It's called sterile insect technology. Or also there's a technology just coming out on the market. It's called transgenic mosquito. It's from Ozitech company. And SIT, you have to release only single sex. Usually with people releasing the male into the environment, and the male close to the wild type female could saturate their mating capacity and turn out could reduce the whole population. And also the same for the transgenic mosquito from Ozitech. They also releasing the male mosquito into the environment, and the progeny are able to hatch. So you have to repeating to doing this, and that means the common feature of this population suppression is the repeating activity costs a lot of money. And once you stop it, and the population will back to the normal. So the other alternative we call is population replacement. So that's the major thing we are talking about is a gene drive. And the other is another technique called Robatia infected mosquito. So it's different from the population suppression. These methods are actually could be self-sustainable. So once you release the mosquito for some time point, after that, you could stop it. And the whole population could be self-propagate and self-sustain to cover the wild type population to become, all the population become a new generation. So we don't need to repeat releasing. So such kind of the substitution after some point and mean it could be more economic. But there's also has some disadvantages for such kind of the strategy. One is you need to release sometime for the both sex, including the male and the female. For the mosquito, releasing the female mosquito, you need a strong community engagement. And the other disadvantage is once the mutation occurs or resistance coming out from the new population, maybe you need to think about how to recover this population in the future. So using the gene drive, we need to achieve the two major points. The first major point is you need to design an effective gene for malaria and the dengue virus resistance. And then you need a drive, that's what we call gene drive here. The driver gene should be suppressing the effective gene into the population and eventually replace it. So the solution had to link the gene for pathogen resistance with the gene drive, and sometimes people also call it the selfish element. So we from the previous first section, we learned what is gene drive. Gene drive means it could against and not follow the traditional Mendelian inheritance. So if people understand the principle of PCR, some people call gene drive as another name as a mutation chain reaction. So the gene drive will contain one cassette. This cassette has the ability to cut off the sister chromosome to making a chromosome break, and using the homologous recombination to repeat the whole cassette into the other chromosome. So we know PCR only require a couple seconds to achieve the reaction, but such kind of gene drive, you need to multiple cycle for the organism life cycle. So you can see, for example, here, the blue gene drive mosquito, they're close to the wild type, and the all progeny are the same as the blue one. So they could spread in their generation, and if we could put the anti-disease cassette into the gene drive, then you could create a new synthetic population which could be disease refractory. So we are talking about how could achieve this point. The first is for malaria. We know the malaria Plasmodium parasite, once the mosquito vector attempt the patient blood, and such kind of parasite will become maturation in the mosquito midgut. And that's a very some dramatic behavior for parasite. They will escape, they will penetrate and go through from the midgut into the hemolymph for mosquito, and finally will infect the salivary gland. And the salivary gland will be another organism for replicate the parasite and to transmit the parasite into another healthy people, and they get sick. So in this study, they're using fudge displays screening. They find out that a small peptide that only contain 12 peptide could blocking the salivary gland epithelial cell as well as the epithelial cell for the midgut. So using the transgenic technique to introduce this kind of small peptide could block in salivary gland and the midgut, the result could show they could successfully block in parasite life cycle and eventually stop the malaria replication in the mosquito body. So for dengue and Zika, the other kind of the mosquito-borne disease, mosquito also will attempt the virus particle from the patient. And the virus go into the midgut also had to replicate in the midgut, and then just like malaria, they will escape from the midgut, going to hemolymph to infect the salivary gland, and the salivary gland have the new virus particle. They will biting the other healthy people and spreading this disease into the population. So our idea is using the shRNA to targeting dengue virus or other vector-borne disease. And maybe we could block in the virus replication in the mosquito gut. So in this scenario, we can using the small shRNA, which could targeting dengue virus, as well as the chikungunya virus, or even we could combine them together to become the two-in-one construct and making a transgenic mosquito. So in this result, you could find the transgenic mosquito compared with the wild type, that has a much low viral load with the regular mosquito. And that could indicating this design actually could become a disease refractory cassette. The same idea also been applied to targeting to Zika virus. So this is the data showing the transgenic anti-Zika small shRNA mosquito, and they also showing the very strong inhibition for Zika infection. And the other is strategies using the monoclonal antibody. This monoclonal antibody, just like Professor Ong-Yong say, the dengue virus had four different types. So they are the common targeting single strand antibody could neutralize 4 different types of the virus. And using the transgenic technique, you could introduce this refractory cassette into the population, and then you could see the mosquito actually has a strong inhibition, not only for one type, and the 4 different types of virus also get inhibited. So if we could put the refractory gene and put into the gene drive system, and you could releasing such kind of mosquito into the wild type, which is the disease transmitted strain. And after some generation, you could see the wild type will be eradicated and all the population will become disease resistant. So here is a 3 take-home message from my section. The one is we do need a new tool for preventing vector-borne disease before the vaccine and the drug come into the market. The second is the gene drive. Once been developed, it will be much less need to repeating releasing. Compared with the original traditional suppression method, it might be economic than the other one. So the gene drive actually are more specific than the other control method. The advantage for this design, they could only aim for the specific species. And I will thank the collaborator from here, from Pasteur France and UCSD. So here is my talk. Thank you.
Thank you very much, Dr. Chen, for that presentation outlining the technology of gene drive and what is required to achieve better disease control. So let's move on to our final speaker before discussion in the webinar today. So we would like to turn to Dr. Maria Vittoria Mancini, who's going to be presenting us with an overview of the potential advantages and challenges associated with using gene drive to control vector-borne diseases. Over to you, Dr. Maria.
Thanks very much. Good morning, everyone, or good afternoon, depending on the time zones. Thanks for having me here today. And as a team member of Target Malaria, the main focus of my work is to develop gene drive technologies for tackling vector-borne diseases. Today, the topic of my talk will be a description and an evaluation of the usefulness and limitations of using gene drive approaches for controlling vector-borne diseases. As the previous speakers already explained, with really nice information and details, vector-borne diseases still represent a major public health burden throughout the world, accounting for more than 17% of all the infectious diseases. And these public burden do not only have profound human costs, but it also restricts the socioeconomic stages and development of countries having the highest infection prevalence. Despite the large gains of using traditional methods of control, the situation and the epidemiological situation of vector-borne diseases, it's currently stalling. So the use of insecticide, bed nets, or indoor residual spraying, they represent good methods, but at the moment they're becoming increasingly insufficient. So the urge.
Apologies to just break in a second. I think we cannot see your screen.
Really?
Could we
sorry.
again to share the screen, please? Thank you.
Of course. Sorry. Sorry
not
about
a problem. I think we just need to put it onto slide mode.
Yeah, sure. Can you see
Perfect.
now?
Thank you.
Sorry. Okay, so I'll just, this is the introduction that just made on vector-borne diseases. So I was saying that yes, we need additional control, effective methods, sustainable methods, and innovative strategies for tackling vector-borne diseases. One such approach is the genetic control, and specifically gene drives. We can use the genetic control for controlling vector-borne diseases, and we could do it by introducing modified mosquitoes into a population in order to reduce the population density and in turn to decrease or even eradicate a disease. There are several genetic traits of mosquitoes that could be of interest for developing specific genetic control methods. For example, we could target the ability of mosquito to transmit diseases. So we could exploit their ability to be refractory to pathogens. We could target their ability to find a blood meal and to get a blood meal, which we know is an essential step during the pathogens transmission and also for the generation of progenies. We could also target female sterility, and we can make these mosquitoes unable to produce viable offspring. Or we could even target sex determination pathways of mosquitoes. So that could be the opportunity to generate mosquitoes whose progeny is only represented by males, which we know are not involved in the transmission of diseases and do not bite. So within the strategy of genetic controls, one technology are gene drives. As explained before, gene drive are selfish genetic elements which are able to induce a bias in the inheritance of genes. The technology exploits some DNA cutting enzymes called nucleases, which are able to modify mosquito genes in a very precise and specific way. So as I said, these mosquitoes are able to skew the inheritance, the inheritance pattern. So we know that most genes are inherited half the time, and they follow the traditional Mendelian inheritance. Gene drive instead are always inherited, and you can imagine that these will give this gene the opportunity to easily spread and within a population and do it relatively quickly. Gene drive can be used for different type of population approaches, and they can support different type of population approaches. So we could use gene drive for suppressing a population by releasing mosquitoes that can cause transient or permanent population suppression, or we could use gene drive for replacing a wild-type population with, for example, mosquitoes which are refractory to pathogens. And gene drive allows both strategies because it supports and it provides the ability to the genetic modification to spread through the population in a very efficient way. So I think that I've already listed a few benefits and promising features of gene drives, but let me go into the details and show you a much more comprehensive scenario of the benefits of this technology. So from the lab evidence and has been collected over the past 10 years, and the predictive mathematical modeling that are assessing the potential of this technology, we know that gene drives do have the potential to reduce mosquito population and to have a substantial epidemiological impact in decreasing the transmission of vector-borne diseases. They are able to reduce the economic and human costs of these diseases, but also to have an impact also at environmental level in controlling invasive species. It's a long-term approach. We know that it's more effective than traditional control strategies like insecticides, but at the same time, it's not mutually exclusive. So it's compatible with other strategies, with even traditional control strategies, and it can work synergistically with other interventions. As I mentioned before, one of the greatest potential of this technique is the fact that it's self-sustainable. So it has the ability to establish in the environment and to spread. And it's widely applicable, meaning that transgenic mosquitoes can reach species distributed in large geographical ranges. Another important feature is its specificity. So it's species specific, and this reduces the undesired effects on non-target organisms compared to other conventional methods. Let's think about like massive insecticide fogging and the effect at ecological level. And it is also accessible. It's accessible in terms of efficacy of the technology because it's effective at low prevalence. So you might need a limited number of initially released mosquito in order to make this mosquito able to spread and to establish in a population. So it has a low frequency invasion threshold. But it is also accessible in terms of cost effectiveness because it's easy to deliver. It does not require major infrastructure improvements or changes, and does not need human behavioral changes in order to be implemented as a strategy. So everything sounds really promising, and everything it's really promising, but this technique and this technology has intrinsic limitations of different types. So the first one, it's really a technical limitation, and it's related to the scientific side of the technology, and it's related to the potential reduced efficacy of the technology, meaning that the technology that we have developed is not as efficient as we thought. There are different aspects related to these, different technical aspects. I've included two of them, which I think are the most meaningful. The first one is the genetic resistance. So gene drive alterations, or any drive-mediated genome alterations, are expected to select for resistance on an evolutionary timescale in different organisms. And these evolution of resistant traits can have the potential of preventing the spread of the driver. But there are already strategies in place. So scientists are working for improving and for decreasing, minimizing the risks of development of genetic resistance. A possibility is to target very highly conserved sites or highly conserved processes in the mosquito biology, in the mosquito physiology, or we could target the sites, the DNA sequences or the processes at different sites, multiple sites, or at multiple stages if we are targeting the pathogen, for example. Another possibility is the fact that gene drive individuals have higher fitness cost compared to wild type. So these mosquitoes, once they will be out in the field, they won't thrive that well compared to wild type mosquitoes. But also here, we have tools in our hands to minimize the possibility of this scenario because we could improve the gene drive technology itself. So in other terms, the genetic construct, or we could improve the target gene that we are targeting. So we could aim for a gene, for example, that has a milder impact on the mosquito biology and physiology. Another set of limitations and challenges for gene drives relates to the public perception and the ethical concerns related to the technology. So there is the possibility that the technology is considered and perceived as an interference with nature. but public engagement efforts and programs and the consultation processes, including transparency and honesty in really identifying the benefits and the burdens and the limits of this technology, could really help in making people, stakeholders, community, and everybody aware of the potential of this technology. And this has to be done together with an informed and structured decision-making process. A second point relates to another aspect of gene drives. As I said, a great potential of the technology is their ability to spread. But at the same time, this could be a limitation, because there is the risk that gene drive will be difficult to stop if potential unexpected effects might occur during the developmental phases. In relation to these, we have to keep in mind that the main aim of scientists and developers and regulators is to have and develop the safest and most effective technology through the application of responsible science, and especially through the application of stepwise approaches and thorough risk assessments for every single step of the developmental, the testing, and potentially of the application of the technology. Other points relate to the lack of adaptive governance. So over the past few years, gene drive research really achieved really incredible progresses, and new aspects of the technology has been discovered and described, and this created a gap in the international legislation. So there is the need of developing guidelines and specific recommendations to regulate the developmental phases of the technology, but also the application of it. And the last point is related also to the need of implementing international legislation, because there is the potential that this mosquito will spread across national borders, and this could result in international regulatory incidents. So also, in this case, the need of implementing international guidelines, it's really a key step for having an effective and a really efficient strategy against vector-borne diseases. A final aspect that could represent a limitation and a concern in relation to gene drive technology is the possibility of ecological impacts, especially when gene drive are applied for population suppression. So the question is, what would have happened if we release gene drive mosquito, we suppress a population? Are we going to disrupt a specific ecosystem, a specific ecological niche? So there are ecological studies at the moment, they are ongoing, they are specifically addressing these questions and these concerns. However, there are evidence, there are showing that, for example, Anopheles gambiae, which is the target organism of target malaria, represent a small fraction of the invertebrate biomass. And it's not known to be a specialist pollinator or a keystone species in the food chain or in the ecosystem, because it represents really a fraction of the ecosystem of mosquitoes and, of course, of insects. And predictions actually suggest that potentially the largest ecological effect that might result after population suppression and reduction of mosquito population could be the reduction of malaria, not so in relation to the disease and not really to the vector. So concluding, I just want to stress the need for innovative control vector interventions, and this urgency is becoming increasingly apparent. However, a key point for developing effective and safe strategies is to include a transparent, inclusive, and well-informed discussion in order to have a responsible evaluation and actual development of this technique. This discussion needs to consider the risks and the benefits of the technology, but at the same time, it has to consider also the benefits of maintaining the status quo, of having a situation where vector-borne diseases still represent a large public health burden. So many thanks for your attention, and I'm happy to take any questions.
Thank you, Dr. Mancini. You've certainly given us a lot to think about. And I think this leads really well into the Q&A section and forthcoming discussion. So a note to all attendees and participants, if you would like to pose a question, please can you use the Q&A feature on Zoom to do so, and we'll go through the questions on the Q&A box and then address them to the panelists. If you have a question in mind for a particular panelist, please can you specify that in your question, otherwise the questions will be posed to all panelists. So I'd like to kick off our discussion at this webinar today with a general question addressed to all panelists. We've just heard about a number of benefits and challenges associated with successfully implementing the gene drive technology. What do you view as perhaps the key barrier to successful implementation? And so I'll go to each of you in turn to see from your perspective and with your expertise, what you view as the key barrier to implementation. Let's start with you, Prof. Ooi.
Thank you. Thanks very much. I guess it's a very elegant technology, and I think the potential certainly is there. There are several issues. One is depending on which model you use. I mean, both Dr. Chen and Dr. and Victoria have basically given an outline of how it can be used, but whether you want to use it as a replacement or a displacement kind of strategy. I think each one has its strengths and weaknesses. One is the cost, right? So if you want to basically just release males and all that, and you keep having to release males even if the disease is coming down, even if the mosquito population is coming down, because if the minute you stop, then that's gonna everything could reverse. Whereas if you have a gene that is driven like Wolbachia, the bacteria that's driven through the entire population and rendered the mosquitoes less susceptible to transmission, then that could be good, but you cannot reverse that once it's gone through into the field. That if there is any issues that arise, that there is no way you can reverse that. Right? So there are pros and cons, and I guess that either one is associated with the cost of the program, that you still have to generate these transgenic or other forms of mosquitoes and release for them to be released in the local area. So yeah, that's just my thoughts on the subject.
Thanks. Dr. Chen, you're on mute at the moment.
So I think vaccine would be the best choice. Once the vaccine been developed, I would say the gene drive probably would not be urgent needed. But the gene drive system actually had some fundamental problem. You know, technical wise, we could achieve some point and could show it could workable, also could have some benefits. But I think the regulation will be the key. You know, the other thing is the engagement. The engagement also involved most of all the different society and also community to agree with the releasing such kind of the animal. So we're talking about the transgenics. Most of the time, right now, we get benefit from the transgenic plant. For example, the corn, rice, or soybean. But mosquito or other insect, they are animal. They are more difficult than a plant for controlling. So the regulation will be a new task for human. You know, although we have some trial, you know, it's been deployed in Africa or some other place, but we still need more data and more evidence to convince people they are actually safe. Just like Anyong saying, once something happen, and how can we do that? If we have the other choice, we need to do using the version 2 to cover version 1 and version 3 to cover version 2 and so on. So that's some fundamental question had to be addressed in the recent years until we could have all people agree, all people feel safe, and that they will be at the maturation time to push onto the market. And that's my point.
Thanks, Dr. Chen. Dr. Mancini.
Yeah, I totally concur with these views. Assuming that we have in our hands the safest and most effective product that we can develop with its regulatory dossier, with a thorough characterization of the risks. So assuming that we have these on our hands, so we're safe and ready to go. I think that the key point is to have the consensus and the cooperation at national level, international level, but also in the local communities. And these has to be based on the rigorous evidence, on the transparency, as I said, on the honesty, and really to have a stepwise approach and a shared decision-making process that definitely shows that the benefits overweights the risks.
Thanks. So in today's talks, we've heard a lot about the application to mosquitoes, and understandably so, given the burden of on morbidity and mortality that diseases like malaria and Zika place. But we've got a question from Sridhar Ranganathan, who is quite excited about the gene drive experience, but is asking, are there parallel opportunities or attempts to try use this novel and effective technology for controlling any of the insect transmitted virus disease or diseases in plants? And it also relates to another question by Babnish Kumar, who asks more specifically, can the same technology be used against desert locusts, so to prevent them from damaging crops? Please go ahead, any of the panelists who any thoughts to share.
Let me first. I think that requires some information. For mosquito, the transgenics technique are beginning from the early 90s. So right now it's about 20 something years. So I think other insects, you need to have how to do the successful rate for the germline injections. And the second is to have the whole genome sequence, because you need to edit in the genome or put something else into their DNA. That's a fundamental knowledge you need to get at the first time. And the second is, for mosquito disease, it's important, but for economically wise speaking, it's also very difficult to become a market, because all the public health are under the bureaucracy of the government. So you're not able to think about, there's a company could control the disease. That's an example for the Ozitech, which is I think maybe the very few number they are running the company focus on the mosquito. And the other thing is, even the Ozitech, they have the technique also applied to the agriculture. So I think gene drive maybe in the future it's a good choice, because it didn't affect to the human health. It's more simple. And if we could have this kind of a disease refractory gene, for example, everything could apply into the plant disease and crop disease. And if the vector insects also could be engineering, I think that there will be one of the direction in the future.
Thanks. Is there any other comments from our panelists on that particular issue?
No, I can add. I guess I agree with Chun Hong that Oxitec has done some of this work before, and I think they've tackled some of the agricultural pests, although not necessarily for virus transmission, just as pests, and they've worked well, right? But at the end of the day, I guess all of this has an economic, it has to be an economic motivation to it, right? Otherwise, the program itself will cost money to generate the transgenic insect, as well as to release and control and monitor. And so there must be sufficient of an economic case for it to be used.
Thanks. So just by surveying the questions in the Q&A, I see we've got a lot of technical questions on implementation, and I guess that's probably the first type of questions that are raised. So here's a question. It's to address to Dr. Chun and Dr. Maria Mancini. How does a laboratory gene drive mosquito, how would that be effective in suppression or replacement in an open setting, in diverse, harsh environments, and that too different parts of the world having different types of environments, even for the same disease, even for malaria, for example? How will they survive, and will the community welcome these gene drive modified vectors? So it's quite a I think a loaded question, but if anyone would like to address any part of that question, please go ahead.
Yeah, maybe I'll just go ahead, because I'm directly involved at the moment in the characterization of and preparation of a regulatory dossier for a specific line. Yeah, so this is actually a really good question, because it's a key point for the effectiveness of the technology. The idea is to there is a technical terms which is called introgression. So we basically introgress the lines that we create in the lab, which are adapted to the conditions of the lab. Of course, we introgress them in the genetic background of the wild type population that we have in the very specific site where we want to release the mosquitoes. So this is a long process, because we need to back cross the genetic backgrounds, and then these lines will be characterized and thoroughly analyzed, and in order to understand the potentiality and the potential risk that might create. So it's a really important point, because it defines the effectiveness of the technologies of the mosquitoes once they will be released in the field.
Thanks. Anything to add from our other panelists? Yes, Dr. Chun. I think you're on mute again.
Okay, so yeah, I agree. Yeah, that's a lot of the factor will be affect. It also very vary, from point to the other point. For example, the insecticide resistance. So we do observe the insecticide resistance is a big factor in Taiwan. For example, we had a city called Kaohsiung that has been spoken for over decades, after World War II. So if you test it, there will be very strong resistance compared with other cities. So even though the cities are not distant so far, not so you could imagine that country to country that there will be a huge difference. So backcross is one choice. You could do that. And the other is the environment, especially, for example, in Robachia project, and the first time deployed in Vietnam, and they engaged some problem because the temperature is too high than Australia. So the population just crashed, after releasing for a couple months. So they had to re-engineering. So it's pretty much like a try and error, although we scientists think we know almost everything, but until you want to release in the real world, there's so many factors you have to concern. So I would say, we could do like to fix all the 1,000 conditions, but once you have to have the semi-field test or some local small region test, they will tell you more factor you have been not really considered about it before. So in that sense, that we always conquer by the try and error. That's it.
Thanks. And so that question, my apologies, was from Dr. Syed Shah Hassan. And it also leads to another implementation question by Nahid Kausar, who says, and it's a point you raised, Dr. Mancini, that mosquitoes don't know borders, they don't know land borders. And so we've just spoken about tailoring the gene drive experience in one setting, yet these mosquitoes can cross borders as well and into a different setting. How do you think we might in the future be able to combat such a challenge? Perhaps I'll just Dr. Mancini first, yeah.
Okay. So as I said, definitely we need to regulate this from the governance point of view, because and this raises the point that we need specific regulations for gene drives in insects, because everything is mostly based on genetic control strategies on crops, which we can easily understand. They stay there, they don't move. Instead, mosquito can fly and can move. So it's different. From the ecological point of view, instead, I mean, we believe that there are studies on the dispersal movement of mosquitoes, so we can characterize the transboundary movements of species, of populations, but at the same time, the technique is very species specific. So it depends on the type of environment where the mosquito, that species of mosquito is specific. So it also related to speciation and to ecological limitations and geographical boundaries. So there are different aspects that could be considered when we assess how far mosquitoes can go.
Thanks. So I'd like to address a question to Dr. Prof. Ooi. And it's in relation that we've spoken about malaria and dengue. We know that there's a global initiative to eliminate malaria by 2030 in the Asia Pacific, but also in Africa and South America as well. With the multi-pronged approach that you raise, and if gene drive were a part of it, on the one hand, we've got a question from Bharat Char, who asks, what would the time scale for successful gene drive implementation need to be in order to actually achieve results? Has there been any modeling done in the area? And on the other hand, with elimination, we're quite close to 2030 in the scheme of things. But even if elimination of malaria is achieved through, say, gene drive treatment and other vector control, what happens thereafter? And this is also now a greater question on cost, because maintenance, countries will be doing elimination one at a time. And so there's going to be maintenance required. What are your thoughts on the matter? Will the time scale that gene drive will take to be implemented fit into these nationwide goals and regional goals? And is it actually going to be necessary even after elimination has been achieved?
Oh, yeah, that's a great question. I guess if I can speak of this from the Wolbachia experience in Yogyakarta. So that's the trial, not clinical, but it's a cluster randomized trial in a city in Indonesia, where the mosquitoes that were infected with Wolbachia and then allowed to introgress into the population. And the strategy was to make these mosquitoes refractory to transmitting dengue virus. So I know that in some detail because I was on the independent data and monitoring committee. And so if you look at how fast the cases come down, I mean, I think within about a year of once enough of the population were infected with Wolbachia, and by that I mean about 30 to 40%. Once that reaches that threshold, it seems to take off on its own. Then you see quite a remarkable drop in the number of cases at the same time. So using a strategy like that, you can kind of see, if nothing changes, like what has happened with the same strategy in northern Queensland, where after 10 years, quite a sizable proportion of the mosquitoes remain infected with Wolbachia, then you can see how this can be sustained, right? But not every country is using this strategy, and as Chun Hong had mentioned, not everywhere will see similar success, right? Yogyakarta in Indonesia is quite high up in terms of altitude. So as you get towards the coast, like what happened with Vietnam, where it's warmer and more humid, then maybe the effectiveness will change. So I don't think that we know enough to be able to say for sure that whatever happened that we see in Yogyakarta will apply to, say, Singapore, to Rio de Janeiro, to Cape Town, to wherever else that you think this is needed. Then, of course, there's the issue of, can you use chemoprophylaxis, so medicines, right, to treat people to tackle malaria, and use vaccine at the same time. Again, I think the theoretically is viable. Practically, I'm not sure that how much, because how compliant people will be. This requires, of course, political commitment and commitment for years, right? So will the various ministries of health see through this program? The cost of the drug has to come from somewhere. Someone's going to have to pay for it, and then someone's going to have to monitor for resistance, and the compliance level that if you give them a pill, they're going to take that pill and not throw it away. So logistically, there's a lot of challenges, and I think theoretically can be done, but it's going to require a lot of political will and discipline from everyone involved, and that part I'm not so sure.
Thanks. So you brought up this issue of resistance, and so there's a question in the Q&A box as well from Dr. Samia Taslim, and raising exactly this idea of can these genetically modified mosquitoes now also become resistant to infection? We've seen this now with drug resistance being quite a problem in malaria and fast evolving, also with insecticide resistance for ITNs, insecticide-treated nets, and IRS. What do we foresee sort of the future to be with respect to resistance and gene drive?
Right. So by that, I mean, I think the question is, you're referring to whether the parasite or the virus will overcome the resistance mechanism. I mean, I think the short answer is yes. I think we should expect it. Everything, if the virus is fit enough, it will evolve and it will overcome, right? And so that's why, regardless of what we use, be it vaccines, be it Wolbachia or other gene drive mechanisms to prevent transmission, there has to be surveillance at the same time, right? And the surveillance cannot be set aside just because the disease is now, on its way down. And actually, all the more the surveillance need to be stepped up so that you can detect problems early and then hit them off before they become too big. So yeah, I mean, the short answer is yes. I think we should expect resistance, and we should watch out for it and plan for it and start thinking about how we're going to deal with it.
Thanks. So a question to all panelists, in reviewing many of the technical and implementation type questions that are being raised in the Q&A, so about application to plants and parallel methods that are being developed, what do you view as perhaps the best way to encourage these discussions and in the scientific community in order to accelerate the development of and implementation of this technology? So, for example, a webinar like this is one method that we are raising these important questions, but this is not necessarily the best forum to answer all of these questions. So, how would you encourage our participants today to become more educated in this field and to stimulate these discussions in their settings as well?
Let me start then.
Thanks.
I think again, coming back to the World Mosquito Program's effort in Yogyakarta, I mean, it wasn't like they decided that they would do that trial and then therefore started. There was a preparatory stage of, and I forget how long it was, maybe more than a year of just, training, having a conversation with the leadership of the city, the mayor and the senior administrators, and then from there filter down all the way to the grassroots, to the citizens in that city, so that at least everyone was aware. Any concerns could be raised, and then we could have a debate, and information could flow, so that at a time when you start this program, at least people are aware what's going on. They're not going to be taken by surprise, and that whatever that they find, it was whenever possible, it was fed back to the population. So there was this very constant two-way conversation. So I guess the long answer to your question is, I think the more conversation we can have at all various different levels, from the scientific discussions all the way to the ethical discussions, the safety, the ecological impact and all that, the more we can debate this then, and as openly as possible. And I think the more people will become aware, and once that happens, and I think the journey will be a lot smoother.
Thanks. So we're fast approaching the end of the Q&A session, and it's been quite a fruitful one. So I'd like to end off on a question, once again, addressed to each of the panelists. And so the COVID-19, this is on our minds all the time. And so what we've seen in COVID-19 is perhaps unprecedented. It is rapid innovation at a pace that is perhaps surprising to all involved in the field. We produced effective vaccines in a matter of months, and there's been a large funding direction in the field of in COVID towards COVID-19. If such attention and investment were directed towards gene drive, how do you think technical progress would be accelerated? So in another way of framing the question, where would you see gene drive with such investment in a couple of years? Where could it be compared to where it is right now? Let's start first in the other order with you, Dr. Mancini.
Thank you. Yes, definitely there would be a great improvement of the redirection of fundings. So for us, it would be great. We will have for us, sorry, scientists, we will have lots of chances and to address questions that we might have in mind. At the same time, we are very well aware that there is a stepwise approach that we have to follow. So now we are characterizing lines, mosquito lines, that potentially could be released in 20 years. Because, as I said, we really want to have in our hands the safest and most effective product that we can have. So yeah, at the same time, it would be really interesting and really helpful to really have fundings for addressing all the possible questions, concern, and also to use these fundings for public engagement, for outreach activity, for really making the discussion inclusive as much as possible at a different level. Community, for example, involved in schools, so that people feel responsible and feel the responsibility of being part of the program, because it directly affects local communities. And at the same time, to have an international and global regulation and governance body that can lead somehow in an organic way, let's say, and between different countries. So yeah, the discussion, I think it's a really key point for us who are developing the technology, for people who are benefit from the technology, and from regulators, from third parties, and leading global organization related to public health. So yeah, I really believe that an inclusive, transparent discussion is a way forward.
Thank you. Dr. Chen, over
I
to
think
you.
the big investment to the vaccine development in COVID-19 outbreaks, probably not also concurrent to the gene drives research, because the vaccinations and the vaccine process has been established for a long time. So only have the one new product and people know how to do it and they know all the out sequence what will be arranged. But for the gene drive, I think there's so many technical gap and also the regulation gap, around there. So even we could have the technology or scientific achievement, but doesn't mean we could have the same success for the Moderna, the BNT, and the vaccine in this scenario. So I would say, they still need some success cases, just like Jakarta or other cases. We should have the more, and we should learn from not only scientific achievement and also for how to improve the social work to convince people the new technology also could be benefit their life. So I would say, for gene drive, they still need longer time than others.
Thank you. Prof. Uy?
Yeah, so I think from the COVID-19 perspective, I think it would seem that the vaccines, were developed in a really world record time, right? But actually, in reality, the mRNA vaccine took about 20 years to develop. I mean, it was way back to how mRNA could be used and how do you package it so that you could deliver it into the body without it having been destroyed before it does what it does. And also we've had 2 outbreaks in the form of SARS and MERS, where a lot of work was done to figure out which part was the best part to go after if we had to develop a vaccine. So it's that body of knowledge that then allowed us to translate and develop these vaccines. So I think I agree with both Maria and Chun Hong, right? That we still need that data. We still need a lot of fundamental studies to be done both in the lab and in the field. And the more we do this, then the better that foundation would be for us to be able to scale it up and do it at a big time, global or regional level. But until then, I think that's therefore that strong case for funding support for fundamental research work that what Maria and Chun Hong are doing.
Thank you. And thank you to all our panelists for a really excellent and vigorous discussion at the webinar today. If we were to look at the key takeaways from our session today, we understand that the race for new therapies and interventions against these vector-borne diseases is almost never ending. And there's a definite need for new tools in order to combat these diseases and achieve the targets set out by the countries around the world. We've learned about how gene drive works and how it can be implemented, and very importantly, the limitations and the benefits of gene drive, and particularly the need for transparent, inclusive, and well-informed discussion on how to do this responsibly and how to achieve good governance. And this is exactly the topic of the next webinar in this gene drive series. So I do encourage all the participants today to attend the following seminar as well. And so to conclude our webinar today, I'd like to invite Dr. Rodora Romero Aldamita, or Ola as we fondly know her from ISA, in order to say a few words. So Dr. Rodora has extensive experience in biotechnology. She's the director of the Southeast Asia Center and director of Global Knowledge Center on Crop Biotechnology of ISA, based in the International Rice Research Institute in the Philippines. She coordinates the activities of ISA's biotechnology information centers in the region, as well as capacity building activities on biotechnology and biosafety in Southeast Asia. So thank you again to all participants, and over to you, Ola.
Hello, everyone. I know that the discussion is really very interesting. I see a lot of questions which were not answered, but we hope that we can answer those as we go to the next one. But before I continue, I'd like to invite everyone to please respond to the post-webinar survey. It's already posted at the chat box. and that all respondents will be able to receive the certificates. A lot of you guys are asking about certificates. So on behalf of Outreach Network for Gene Drive Research, the ISA-BICS, who are joining us now, and also ISA, we would like to thank all the panelists, Dr. Eng Ong Uy, Dr. Chun Hong Chen, and Dr. Maria Vittoria Mancini for their comprehensive presentation and for engaging conversation during the Q&A. It's really fantastic. Our moderator, Dr. Sheetal Silal, for her excellent facilitation of the webinar, her additional comments during the Q&A, and for giving us a summary of the whole webinar in the form of key takeaways, and of course, relating Gene Drive to our COVID pandemic currently. We would also like to thank the active and excellent participation of our audience. A lot of questions, as I said. So from today's webinar, we learned that Gene Drive is an important tool in preventing vector-borne diseases. It is species-specific, and once developed, there is less need for repeating re-release. In order to make informed choice, a transparent, inclusive, and well-informed discussion should take place, and risk and benefits of the technology should be determined before the technology is adopted. ISA supports the understanding and application of Gene Drive technology as we see its many potential contributions to food security, environmental sustainability, and in the improvement in human health, which we have learned today for dengue, chikungunya, Wolbachia, and malaria. So as ISA's facilitation of information on education and biotechnology extends to other sciences such as Gene Drive, we help prepare the general public and various stakeholders in the acceptance of the technology for the need of the global community. Let us then continue to be educated. We still have webinar number 4. This is going to be on July 22 at the same time, and it is titled Regulatory and Governance Considerations for Gene Drive Research. You can see that. You can now start registering. There is, again, a pre-webinar survey on that, and we'd like to see you all once again. Thank you so much, and have a great day. Bye-bye.